We'll discuss some of the mechanisms of cell transport now. Cells are selectively permeable because the membrane allows certain molecules to pass through it, but not others. This ability is critical because it allows the inside of the cell to differ from the outside environment, and by extension, allows the inside of the body to differ from the outside environment. There are two main mechanisms for molecules to move across a cell membrane. The first is passive transport, which requires no energy, and active transport, which requires energy. Passive transport does not require energy input because it moves a substance from high to low concentration, which occurs naturally due to kinetic energy of the particles. Methods of passive transport across a membrane include simple diffusion, facilitated diffusion, or osmosis, and we'll go through each of these methods in more detail. This figure shows how diffusion works when a solute is released in a media like air or water. In the left panel, area A has more solute or dissolved components than area B, so there's net movement of solute from area A to area B. Net movement of solute continues until the concentrations are equalized between the areas. When this happens, it is in a state of dynamic equilibrium. Movement of solute still occurs, just not in any overall direction like before. This process is called diffusion, and it occurs naturally when a concentration gradient exists and the molecules involved are in a media like air or water. Note that we do not have a cell membrane in this figure. A cell membrane has been added to this figure. A concentration gradient is evident on the left, and the passage of time is shown at the bottom of the figure. Simple diffusion involves the movement of solutes directly through the lipid bilayer from an area of high concentration to an area of low concentration. In this example, the solute starts out at high concentration in the extracellular space. Then there's net movement across the membrane into the cell. Over time, a dynamic equilibrium is reached. Only particular types of molecules can move directly through a cell membrane like this. In order to do so, the molecule needs to be lipid soluble or nonpolar or small. Polarity is the most important determinant for simple diffusion because small charged ions like sodium cannot cross the bilayer easily, while some large nonpolar molecules like steroid hormones can easily diffuse directly through the bilayer. The rate of diffusion across membranes is defined by Fick's law of diffusion. The rate of diffusion, J here, is equal to D, which is the diffusion coefficient, times A, the surface area for diffusion, times the concentration gradient divided by X, or the distance separating the areas of high and low concentration. The diffusion coefficient value depends on temperature, lipid solubility, which depends on polarity, and the size of the solute. Take a moment to look at the equation and think about different conditions that might increase the diffusion rate. Lipid solubility depends mostly on a molecule's charge and can be quantified by its partition coefficient, or Kd. Kd is equal to the concentration of a substance found in the organic indicated by C sub O in the equation versus the aqueous phases indicated by C sub A. Oil and water don't mix when combined and therefore are known as two immiscible solvents. Oil is lighter than water and will float on top of the water in a separatory funnel like is shown here. To measure Kd, you'd add the compound in question to the two immiscible solvents, mix it up, and allow it to come to equilibrium. Then you'd measure the concentration of the compound in each phase. If a compound has a strong charge, then most of it will be found in the aqueous layer and very little of it in the organic layer. Filling in the Kd equation, we see that we would have a small number divided by a large number, and so Kd will be small. If, however, we have a nonpolar compound, most of it will be found in the organic phase and Kd will be large. So a large Kd means that the compound has high lipid solubility. This graph shows the relationship between the partition coefficient Kd on the x-axis and permeability on the y-axis. Note that permeability is measured as distance traveled per hour. Larger numbers on the scale indicate faster speeds, just miles per hour in a car. As you can see, compounds with larger KDs can move through the membrane more quickly. Now that we've gone over lipid solubility, let's go back to Fick's equation and look at different factors that influence the rate of diffusion. Remember that D, the diffusion coefficient, is influenced by the lipid solubility and size of the solute. We've added another equation that shows that the diffusion coefficient is proportional to a compound's lipid solubility, beta, divided by the square root of its molecular weight. Conditions that would make D larger would increase the rate of diffusion. Temperature can also influence diffusion rate. Think about how temperature affects kinetic energy of molecules. Look at each factor listed in this table and, using the equations, predict its influence on the diffusion rate. Here are the answers, and see if you are correct. If a substance needs to move across a membrane via simple diffusion, then modifications can be made to increase diffusion rates, like decreasing the diffusional distance or increasing the surface area for diffusion. But simple diffusion is relatively slow, especially at low concentrations. This fact is illustrated in this graph. We have increasing solute concentration on the x-axis and rate of transport across a cell membrane on the y-axis. We can see from the red line for simple diffusion that as solute concentration increases, then rate of transport increases. But the line is shallow, and concentration needs to be fairly high before diffusion rates increase substantially. These concentrations are not always physiologically possible. So if we want to move a solute quickly at relatively low concentrations, then we need help or facilitation. We can see that with the facilitated diffusion line, which is green, that a substance can be transported relatively quickly across a membrane even at low solute concentrations. But we also see that transport rates level off, and even with increasing solute concentration, transport rates don't increase much anymore. So how is facilitation accomplished? The answer is that facilitation uses membrane proteins to move molecules across the membrane. The molecules move down its electrochemical gradient. And two types of proteins are used. One is a protein channel, which forms a pore in the membrane, like is depicted here. A second type of protein used is called a carrier protein. In this case, the diffusing substance temporarily binds with a specific carrier protein, which causes a conformational change in that protein that moves the substance across the membrane and down its electrochemical gradient. Note that channels are always used in facilitated diffusion because channels form pores or holes in the membrane that allow particular molecules to pass through, while carriers are used in both passive and active transport processes. Carriers are usually called pumps when they are used for active transport. Channels and carriers don't allow all types of molecules to pass through them. Rather, they are usually highly selective and only allow one or two different ions or molecules to pass through them. Here we see a potassium channel. It has a selectivity filter that's shown in yellow. This filter will only let potassium ions pass through it. A substance of a different size or charge will not be able to pass through. Although channels are technically open holes in the membrane, they won't let all solutes pass through them. That is, they are selective. Here's another look at the selectivity filter of an ion channel. And this one allows molecule A to pass through the ion filter that's in the channel pore. and that pore is indicated by the letter B. We can see that the ion filter has projections into the pore that an ion must interact with in order to pass through the channel. The ion must be the right size and have the correct charge to pass through the filter. It makes intuitive sense that an ion that's larger than A would not be able to get through the channel. But what about an ion with the same charge that is smaller than A? Could it get through? This shows a top-down view of the ion selectivity filter in a potassium channel. Sodium ions have the same charge, +1, as potassium ions, but they are smaller than potassium. On the left, we see that a potassium ion is the same distance from each of the oxygen atoms lining the channel's filter. This allows the ion to enter the filter. The arrows indicate that the ion can pass freely through the channel in either direction. But on the right, the smaller sodium ions do not fit properly between the oxygen atoms in the filter. Because they do not interact with the oxygens properly, these sodium atoms cannot pass through the filter, and the arrows indicate that any sodium ions that begin to enter the channel will be kicked back out. This is illustrated by the wide up arrow and the dashed down arrow. We should note that this is how a highly specific filter works. Some channels allow multiple types of ions to pass through them. For example, we'll discuss channels that allow both sodium and potassium to pass through them later, but the principle remains the same. The material that passes through the filter must interact properly with the molecules lining the pore in order to pass through that selectivity filter. Channels can be characterized by the material allowed to pass through them, like the potassium channel in the previous slide, but they can also be characterized by their structure. Gated channels have an open and closed state. Usually they are closed and must receive the correct signal in order to open. This adds a layer of control over what can enter or exit a cell because passage will only be permitted when the correct signal is received by the cell. There are many different types of gated channels, and we'll go through some of them in the next few slides. The first type we'll discuss is a voltage-gated channel. Note that gated channels are named after the signal that needs to be received in order to open the gate. Here, we see that the channel pore or gate is closed. Also note the difference in charge on either side of the cell membrane. The extracellular side is positively charged while the intracellular side is negatively charged. Now the charges on either side of the membrane have changed. This change in the voltage on either side of the membrane triggers the opening of the channel gate. Here's a stretch-gated channel, which is also known as a tension-gated or mechanically gated channel. As the name implies, this channel will open when the cell membrane is stretched or moved in some way. Note that this channel is associated with the cell's cytoskeleton. When these cytoskeletal elements are stretched, this opens the channel. Here's a phosphorylation gated channel. I bet you can guess what signal must be received to open this channel. When the channel is phosphorylated or has a phosphate group added to it, then this opens the gate. The final type of gated channel we'll discuss is a ligand gated channel. When the appropriate ligand binds to the receptor site, the channel opens. So this type of gated channel is also called a receptor. This figure from your text summarizes all of the gated channel types we just went through. The previous slides were animated, so they may be hard to interpret in the PDF of this lecture. So the previous 4 slides are available as a PowerPoint file on Moodle. Channels can also be leaky, meaning that they are always open. These allow molecules to move across the membrane faster than simple diffusion would allow, and these types of channels are typically used to maintain the ionic and osmotic balance of cells. The final method of passive transport we'll go through is osmosis. Osmosis specifically refers to the movement of water across a membrane. Water moves from an area of low solute or relatively high water concentration to an area of high solute or relatively low water concentration. The driving force for water movement is the water concentration gradient. Imagine a beaker of water divided by a semi-permeable membrane, like a cell membrane. A solute is only on one side, side 2 in this case, of the membrane, and it cannot pass through the membrane. However, water can pass through this membrane, and the solute concentration is higher on side 2. Initially, water moves from where it is high to the area where it is low, moving from side 1 to side 2. At equilibrium, there is no net movement of water, and we see that side 2 has a greater volume than side 1 now. Hydrostatic pressure, or the pressure water puts on the walls of its container, opposes osmosis. Osmotic pressure is the pressure required to stop the osmotic flow, and osmotic pressure is proportional to the concentration of non-penetrating solutes. The greater the solute difference between the two sides, the greater the osmotic pressure and the greater the driving force to move water. Does water diffuse directly through the lipid bilayer through simple diffusion? Water is small, but it is also polar. We know that polar molecules generally have small partition coefficients, so they should not pass through the membrane easily. So simple diffusion of water across the membrane is most likely a minor osmotic mechanism. Most water diffuses through channels. You can think of it as a special type of facilitated diffusion. These channels are called aquaporins, and they may be leaky or gated. If gated, they are usually ligand-gated channels. Dr. Peter Agrawal from Johns Hopkins won the Nobel Prize in Chemistry for his work with aquaporins in 2003. Researchers had long proposed that water channels must exist in cells, but no one had been able to find the protein channel responsible. Without evidence of its existence, it was assumed that water diffused directly through the cell membrane, which is what I was told when I was an undergraduate biology student. Dr. Agrawal studied membrane proteins in red blood cells and kidneys. These cells have very different morphologies and functions, but both need to move water across their membranes. Red blood cells need the ability to move water because the body's hydration level can change quickly, which changes the amount of water present in circulating blood. And kidneys are involved in creating urine, which is one of the main ways the body can homeostatically control the body's water content. Dr. Agrawal found one unidentified protein in both of these cell types and hypothesized that this was the water channel. His group isolated the membrane protein and then figured out its amino acid sequence. From the amino acid sequence, they could determine the DNA sequence. And once they knew that DNA sequence, they could then find the chromosomal location of the gene that encodes it. You can follow the link here to learn more about Agrawal's work. We'll go through it in a bit more detail now. Once Agrawal found the proposed water channel gene, the researchers could experimentally block production of the protein. Agrawal had two groups of cells. One had normal cells with the intact proposed water channel, and the other group was cells that had the production of the proposed channel blocked. These two groups of cells were then dropped into a dilute solution. Because the solution was dilute, water should move from the outside to the inside of the cells or toward the higher solute concentration. We see the results of Agrawal's experiment here. Over time, the cells with the aquaporins allowed water to move across the cell membrane, and we see that the cell begins to swell and change shape. Nothing happened to the cells with the blocked protein, indicating that water could not move across that cell membrane. McGraw also looked at a cell that under normal conditions did not express the proposed aquaporin gene. In this case, he looked at the oocytes from Xenopus frogs. He experimentally induced the oocytes to produce the protein by injecting messenger RNA into the cell. The oocyte then had the instructions to produce the protein. The oocytes could then be incubated in a dilute solution, and the movement of water into or out of the cells could be monitored. So what do you think happened? The water could move across the cell membrane in these experimental cells. And this cartoon shows an overview of McGraw's second experiment and its results. The oocytes with the induced aquaporin protein allowed the water to enter the cell, while the normal oocytes did not. Dr. McGraw's experiments provided definitive evidence that aquaporins existed, and they've since been found in the cells of all living organisms. McGraw's experiments relied on the tonicity of the solution that his cells were put into. Tonicity is a measure of the effective osmotic pressure gradient. As an example, red blood cells have a lot of aquaporins in their membranes, and the osmolarity, the measure of solute concentration of a typical human red blood cell, is about 300 milliosmoles. If the red blood cell is dropped into a 300 milliosmolar or isotonic solution, then there is no driving force to move the water across the membrane, and the cell looks normal. If you drop the red blood cell into a hypotonic solution or a more dilute solution than the inside of the cell, then water moves into the cell, causing it to swell. You can remember this by the phrase hypo-hippo. Hippos are bulky animals, so if you drop a cell into a hypotonic solution, the cell becomes bulky, like a hippo. If the cell is dropped into a solution that has more solutes or less water than the inside of the cell, then water is driven out of the cell and into the surrounding solution, causing the cell to shrivel. Because aquaporins are specific to water molecules, they could be used for water purification. A video about the biomimetic application of aquaporins to water purification is available to you on Moodle. The link in this slide will take you to a company using aquaporin technology to create the products you see here. Since aquaporins are virtually 100% selective for water molecules only, these membranes are really effective at removing contaminants. The membrane shown on the top is for industrial applications, while the bottom product pre-filters tap water coming into homes. Most home water filters cannot remove all contaminants, particularly pharmaceuticals. Water in the U.S. is known to be contaminated with these, which is why opiate use in some areas can be tracked through its concentration in those water systems. Osmolarity depends on the colligative properties of solutions, or the number of dissolved entities per unit of volume, and not the chemical nature per se of the dissolved entities. You can think of it as a ratio of solute to solvent molecules. 1 osmol equals 1 Avogadro's number of dissolved entities per liter, or 6.02 times 10 to the 23rd molecules or atoms per liter. Let's look at different solutions found on different sides of a semi-permeable membrane. The solutes cannot pass, but water may pass through, and the volume is the same on either side of the membrane. Glucose and fructose are both monosaccharides. Which way will the water go in this first scenario? From A to B, from B to A, or will there be no net movement? Glucose and fructose have the same molecular formula, but different structures. In this case, their concentrations are the same, so there is no net movement of water. What about this situation? Solution B has twice as many dissolved particles as side A. So water will move from where it is high, side A, to side B. What about this situation? Sucrose is about twice the size of glucose because it is a disaccharide composed of glucose and fructose. Remember, osmolarity depends only on the absolute number of dissolved entities, and side A has twice as many dissolved entities as side B, so water will move from B to A. Here is the last scenario. In this case, the concentrations are the same, but what do we know about sodium chloride when it is dissolved in water? It ionizes and you get sodium and chloride ions. In this case, solution B has twice as many dissolved entities, or higher solute, than solution A, and water will move from A, high water, to B, where water is low. Here are the solutions to the scenarios presented in the previous slide. In all cases, water moves down its concentration gradient to where solute concentration is higher. We finished discussing the different mechanisms of passive transport, and we'll move on to active transport next.